topic is RBC inclusions, one of the most important topic. So when you take inclusions in RBCs, first you have to look what stain is it, okay? So usually the stain done in a peripheral smear is a Leishman stain, which looks like this, in this color, little bit of a pinkish hue. The other stain is a supravital stain, which looks blue in color, which we use to assess the reticulocyte count. Okay, so look at the color of the stain, whether it's a Leishman stain or it's a supravital stain. Okay, now if you have a supravital stain, most commonly supravital stain is done to assess the hemoglobin inclusions in the RBC. So what are the hemoglobin inclusions in the RBC? You have two things. One is Heinz body, the other one is HbH inclusion. So if you have a single inclusion, it's a Heinz body. If you have multiple inclusion, it's called HbH inclusion. Okay. So multiple inclusions will create a golf ball like appearance in the RBC. So golf ball like inclusions in RBC is seen in HbH disease. So what is HbH disease? So HbH disease is a variant it's a type in alpha thalassemia. So you have 4 genes coding for alpha globin gene. So if you have 3 genes deleted, you can get HbH disease. Okay. But if you have 4 genes deleted, you get something called hydrops fetalis. Okay. So HbH disease is alpha thalassemia in which you have 3 alpha genes deleted. Next, when you see a Leishman stain, if you have a single inclusion, it's usually a Howell-Jolly bodies, which is a denatured DNA. So when you have something inclusion or anything to be removed, which is unnecessary in the RBC, it's done by spleen. So when can you have this inclusion in the RBC? When your spleen is not able to remove or if it is produced excessively. So that happens in hemolytic anemia and hyposplenism or asplenism. Next is you have a granular inclusion, little bit of granular, not like a very defined inclusion. A granular inclusion in a Leishman stain is a Pappenheimer body, which are nothing but iron granules, which can be seen in sideroblastic anemia or thalassemia. Next, if you see an RBC with basophilic stipples, very small fine dots, then we call it as basophilic stippling. So those are nothing but the ribosomes which are present, which can be seen in lead poisoning, sometimes in megaloblastic anemia and sickle cell anemia. Okay, so look for the stain. If it's a supravital stain, it's either Heinz body or HbH. You can remember hemoglobin inclusion, 2H, Heinz body and HbH inclusion disease. If it's single, it's Heinz body. If it's multiple, it's an HbH inclusion. And if you have a Leishman stain, look if it is a single or it is a granular. If it's granular, it's a Pappenheimer. Further, you can confirm it by a Prussian blue stain. If it's a single inclusion, it's a Howell-Jolly bodies. If you have multiple small dots, it's a basophilic stippling. Now look at this one. This is a what type of stain? It's a Leishman stain. So Leishman stain, single dot, very well defined, not granular. It's a Howell-Jolly body. Okay, it's a Howell-Jolly body. Next is a supravital stain, multiple inclusion. It's a HbH inclusion, which is a denatured hemoglobin. So what is this? You have Leishman stain, multiple dots, which is a basophilic stippling. What is this? This is a Leishman stain in which you see something like a figure of 8. This is called Cabot ring, which can be seen in megaloblastic anemia. I hope this is clear. This is a very important topic for any entrance exam. So they ask Heinz body is what? It's a denatured hemoglobin. Pappenheimer body is iron deposit. Basophilic stippling is ribosomes. Okay. So thank you. So this is about the RBC.